Welcome to botany class. In the previous class, we discussed crossing over in brief. In this class, we are going to discuss three different topics. Let's start from self-sterility, self-sterility in nicotiana, second topic, sex determination, in maize, and mutation. These are the learning objectives: self-sterility in nicotiana, sex determination in maize, and mutation. Start from self-sterility in nicotiana. In plants, multiple alleles reported in self-sterility or self-incompatibility. Self-sterility. Self-sterility is otherwise called self-incompatibility. What's this? Self-incompatibility or self-sterility. In plants, pollination takes place. After pollination, the pollen grain germinates. Self-incompatibility is in a plant the pollen grain do not germinate, or fertilization, it's not able to fertilize in the ovule. Already you learned this topic in the first chapter very detailed. See this stigmatic surface here. Normally in the plants, this is the stigma, stigmatic region, stigma, style, and the ovary. This is the stigma, this is style and ovary. What happens normally? The pollen grains are dusted on the stigmatic surface. When the pollen grains are dusted here, pollen grain. When the pollen grains are dusted on the stigmatic surface, this is the ovary, the pistil. In the ovary, there are regions, embryo sac, here 3 antipodals, egg and synergid, and polar nuclei or secondary nuclei is present here, covered by integuments. What happens when the pollen grains are dusted on the stigmatic surface? The pollen tube starts to germinate and it fuses with egg. This is the normal fertilization process. In self-incompatibility or self-sterility, what happens during this? This pollen tube do not develop normally. The normal pollen tube development is affected in self-incompatibility. This was reported by yeast. Self-sterility in nicotiana is reported by yeast. In nicotiana plant in the year 1925. So, yeast reported self-sterility in nicotiana plant. One word question. Who discovered or who explained self-sterility in Nicotiana East in the year 1925? So, first you write about the definition, what is self-incompatibility or self-sterility. The pollen grain in a plant do not germinate on its own stigmatic surface. Pollen grain do not germinate the stigmatic surface of the own plant. It do not germinate. This pollen grain, it do not germinate, is called self-incompatibility. So, in Nicotiana, East observed the gene, the self-sterile gene is denoted or designated as S. So, this S, which contains different combinations like S1, S2, S3, S4 and S5. So, different allelic forms. The gene S, which contains different allelic forms S1, S1, S2, S2, S3, S4, S5, different forms. So, here in this cross fertilization, when a cross is made with different combinations, if it is homozygous condition, what is meant by homozygous? This combination with S1 or S2. So, homozygous condition, it do not produces the pollen tube. So, different combinations, look at this tablet column. Here it is selected different combinations of allelic forms, different combinations of progeny in self-incompatibility in Nicotiana plant, Nicotiana tobacco plant. In the tobacco plant, the parents are taken as female parent in the stigma spot S1, S2, S2, S3 and S3, S4. So, homozygous form S1, S1 or S2, S2 which will not produces, it creates self-sterile. But if it is in heterozygous condition, that means S1, S2, S3, S3, S4 or S5, S6, heterozygous forms, it produces the progeny different and the pollen grains are germinate, the pollen grain starts to germinate. So, let us see this. Here in this, if the cross is made between S1, S2 with various S1, S2 made crosses, different combinations are taking place. The parent stigmatic spot S1, S2 in the female parent S2, S3, S3, S4 and the male parent source, it is also S1, S2, S2, S3, S3, S4. Wherever the homozygosity occurs S1, S2 and S1, S2, let us see that how the combinations are taking place. So, in S1, S2 stigmatic surface, in the stigmatic surface is or the female surface is selected as S1, S2. Here this is S1, S2, S1, S2, S1, S2. This is the female or pistil or this parent stigmatic spot. Stigmatic spot is taken as S1, S2 and make a cross between, in this S1, S2, different cross fertilization when it makes, for example, here S3, S4, S3, S4 or it is S3, S2 or S2, S1. These are the pollens, pollen grains. This is the stigmatic region, this is the pistil and these are the pollen grains, different combinations of pollen. What is the condition? In homozygosity, the pollen do not germinate. Other than this, other than S1, S2, it gives the pollen germinate. So, first the S3, S4, see here it is heterozygous condition, here it is heterozygous, S1, S2, S3, S4. So, here the pollen grain germinates, because it is heterozygous condition. You remember that in heterozygous condition the pollen grain germinates. If it is homozygous condition, it do not germinate. Then here look at this, S1, S2, here S1, S3 starts to germinate, but this S2, it do not germinate. Then here S2, S1, S2. So, both homozygous S1, S2, homozygous, in this condition it never germinate. The pollen tube never develops, or the pollen grain do not germinate. Now, look at this tablet column. What are the different combinations of progeny developed in self-incompatibility? So, female parent stigmatic spot, in the stigmatic spot S1, S2, and male part S1, S2, S2, S3, S3, S4. When you make a cross between this, see here is a homozygous condition S1, S2, S1, S2. So, definitely it produces self-sterile. If the pollen do not germinate, what's the thing? It is producing sterile plant. It develops the progeny sterile, self-sterile, because it is homozygous condition. Homozygous condition sterile, heterozygous it develops the pollen tube develops. If the pollen tube do not develop, it do not carry the male gamete, and the gamete do not fuses with the egg. So, it affects the fertilization also. So, it produces male sterility. It produces sterile plant, self-sterile plant. So, self-sterile, here it is self-sterile, and in this combination S1, S2 and S2, S3. So, if it is S1, S2, and so crosses made between S1, S2 and S3, S4. This is heterozygous. See, in this region here and here, it produces 4 different progeny, 4 different combinations. Why? Because the combinations are here S1, S2 and S3, S4. This is S3, S4, S1, S2. Here S1, S2, S3, S4. Here female is S1, S2, male is S3, S4. Here female is S3, S4, male is S1, S2. So, in both the cases it is cross between, this is cross heterozygous condition, cross between S1, S2 and S3, S4. So, it produces, what are the combinations possible? S1, S3, S1, S4, S2, S3, S2, S4. The same it is given in the form of tablet column. The different combinations, what are the different forms? Hope that you understand very clear. The S1 and S3, S1, S3, S1, S4. When you write it here, from the allelic forms taken out the gametes, the possible gametes are S1, S3, here S1, S4, S2, S3 and S2, S4. So, here it is not S1, S2. So, if it is S1, S2, it shows male self-sterility. But here S1, S3, S1, S4, S2, S3, S2, S4. So it gives all the 4 different combinations. Other than this S3, S4, other than S3, S4, S3, S4 wherever present, it starts to germinate, the pollen grain germinate. But S1 and S2, if S1 and S2 is there, because the stigmatic surface is S1, S2, so homozygosity occurs, it do not develop or germinate the pollen tube. This is the condition you studied here in nicotiana. It was observed by East in the year 1925. So once again you look at this tablet column. Here S1, S2 and S1, S2, female and male, this is the stigma spot, this is the pollen source. So it produces S1, S2 self-sterile, and here it gives S1, S2 is sterile. So it is not mentioned here. S1, S3 and S3, S1, this is S1, and S2, S3. It produces 2 combinations. Then this one S2, S3 and S1, S2. So it gives S1, S2, S1, S3, and remaining S1, S2 combinations are sterile. So it is not mentioned. Then this condition S2, S3 and S2, S3. So it is homozygous condition. The homozygous, homo means same condition. So self-sterile. And here S2, S3 with S3, S4, it gives S4, S2 and S4, S3. Then S3, S4, it gives 4 combination. Other than this, S2, S3, S2, S4. Then this condition S3, S4 and S3, S4 also self-sterile. So these 3 are self-sterile because it comes under homozygosity. Then second question. So first question, today's class, first question, you are going to learn self-sterility in nicotiana. Draw this diagram. This all diagrams you draw it in the class note very neatly, and you learn this question. Prepare it well. Second question, sex determination in maize. In maize, the sex is determined. You know that in the first chapter, again you learned this question. Maize. Maize is a monoecious plant. What's meant by monoecious? The male and female inflorescence develop in the same plant. Have you remembered this maize plant? I hope that you remembered the first lesson because you wrote the test also. In the first lesson, you draw this same diagram, maize plant. The maize plant, it develops the monoecious, unisexual flowers. Male flower and the female flower are separate, it is not bisexual. The same plant which produces male and female inflorescence. The terminal region or the shoot apices in the stalk, the terminal region of this stalk, it produces the male flower, that is tassel. This is the male flower, tassel. And the female flower develops at the lateral side. The terminal side, the male flower, male inflorescence develops. Female flower develops at the basal. This is the leaf. Here it produces the inflorescence. Female inflorescence, silk. Silk is developed here. This female inflorescence is named as silk or cob or ear. Three different names for this female flower. What are the different names? The different names are silk, cob and ear. Female flower has these three names, silk, cob and ear. This is tassel. Tassel is the male inflorescence. So, monoecious plant, monoecious, the maize plant, this is the maize plant, XII maize. XII maize. In XII maize, the terminal region, it develops the male flower, and the lateral side it develops the female flower. This is the female flower, monoecious plant. Here there are two different genotypes. The question is sex determination in maize, that is the unisexuality is determined by two different genes, that is BA which represent barren, and tassel inflorescence which represent TS, tassel, BA and TS. So the allelic forms or the gene is BA and TS. The allelic forms are BA, TS, TS. Also you remember here normally the dominant character, it will be denoted as capital B, capital letters and small letters. Here in this cross we are using dominant characters as superscript as TS plus, like that it is denoted as the dominant nature. What are the different combinations and how the sex is determined? So, modifications. In this unisexuality which is determined by the abortion of the stamens in the silk and pistil in the tassel. See here it is unisexual flowers. So, how the sex is determined? By the abortion of stamens in the tassel. So, stamens abort in the silk or ear or cob inflorescence. Likewise, the pistil is abort in the tassel. So, different combination. Now, look at this tablet column. Sex determination in maize. How the sex is determined, male, female. These are the genotypes. There are different genotypes. BA. So, already I told you BA represent barren and TS that is because of tassel. You remember the tassel, the name already you know this name because you learned in the first chapter, tassel. So, BA and TS. The genotype is BA, TS. So, BA, small BA, these two are double recessive, barren and tassel, double recessive. Here this, there is no plus, there is no dominant, this is recessive character, double recessive, dominant and recessive. If the genotype is in this form, double recessive form, what is the modification occurs? It lacks silk on the stalk. So, this is the silk female inflorescence. So, in this genotypic form, double recessive form, it lacks or eliminate the silk inflorescence, and silk is eliminated in the stalk and it is transformed the tassel into pistil. Transformation of tassel to pistil. So, it determines rudimentary female. Rudimentary female develops. What's the modification? It eliminates the silk, but rudimentary, because of eliminating the silk, Silk is this inflorescence, female inflorescence. So, it produces rudimentary female. So, sex is determined as rudimentary female in this genotype. What's the genotype? BA recessive, TS recessive. Then, second genotype. There are 4 different genotypic forms are explained here. So, first one, double recessive. What's the double recessive? BA barren and TS. So, it modifies. What modification occurs? It eliminates silk and the tassel transform the tassel to pistil. It eliminates silk at first and tassel is transformed to pistil. So, it produces a rudimentary female flower. The second genotype is BA and down. BA recessive, barren recessive and TS dominant. TS dominant. So, what happens? Recessive and dominant. Barren recessive BA and TS dominant. So, what modification occurs? It lacks silk and have tassel. It lacks BA is recessive. So, it lacks. Pistil is lacking, but dominant character tassel. So, it expresses dominant character. Here it express male. So, the sex is determined as male. Then, third genotype, BA dominant, barren dominant and both are dominant. TS dominant, BA dominant. So, both are dominant. If both are dominant, there is double dominant. So, both tassel and silk develops. Both tassel and silk develop. So, it gives monoecious condition like this. So, this is monoecious condition, tassel and silk is formed. So, here this is the genotypic expression, BA dominant, TS dominant. So, if both are dominant, it produces the monoecious plant. So, both tassel and silk is present in the same plant. So, what is the condition monoecious? Monoecious is 2 more question. Male inflorescence and the female inflorescence developed in the same stalk or same plant is called monoecious. And finally, what is the other genotype? BA dominant plus and TS recessive. If it is recessive, dominant and recessive. So, first genotype also that is double recessive. Here one dominant, one recessive. Here one dominant, one recessive. It produces the male, it produces the female. Look at these two. Compare this one and this two. Here BA barren recessive, TS dominant. But here barren dominant, TS recessive. If TS dominant, it produces male. Here BA dominant, so it bears cob. So, female inflorescence. The sex is determined as female. Bears cob and it lacks tassel, because that you know, if dominant character is present, it expresses dominant. The recessive, it lacks. Here the recessive is TS, which lacks, and it expresses the sex as female. Hope that you understand this. Once again, you look at this tablet column. There is a 4 different genotype. So, these 2 genotypic forms you learned today. Don't confuse with this. First question, self-sterility, the genus S1, S2, S3, S4. Here, second question, sex determination in maize, which contains the genotype BA, TS. Dominant character is expressed as plus, the superscript. Look at this tablet column. sex determination in maize plant, Zea maize. BA, TS, TS. If the condition is double recessive, dominant and recessive, this is the dominant and recessive nature of the allele, what modification takes place and what kind of sex it determines. If it is double recessive, both are recessive forms, it lacks silk on the stalk and transformed this tassel to pistil. So, it produces rudimentary female. So, expression is rudimentary female. And second genotype, BA with dominant TS. Recessive BA with dominant TS, it lacks silk and have tassel. So, this genotype is familiar to you. If dominant is expressed, dominant and recessive is present, it expresses the dominant character. Here dominant is TS, so it expresses male. Likewise, this one, last one, the BA dominant, BA plus and the recessive TS. So, it bears cob and it lacks tassel because tassel is recessive form. So, the expression is female. And here one, double dominant, both BA plus, TS plus. So, when monoecious condition occurs, so one word question, when monoecious occurs because of the presence of double dominance. So, double dominance, it produces what modification? Both tassel and silk. Tassel and silk produced. So, it produces monoecious. This condition occurs in the genotype. One more question, mutation. You know the definition, what is mutation? Mutation is the sudden change. So, genotypic variations among individuals, genotypic variations. There are different types of mutations, but today we can see the introduction of mutation. You know the definition, already you learned in lower classes. What is mutation or define mutation? Sudden change in the nucleotide sequences. You know the chromosome. In the chromosome, here this is the chromosome. It's made up of DNA sequences. If any change, the genes are located within this, the genes are located in different locus in linear fashion. So, number of genes are located in the chromosome. So, it is in the form of nucleotide sequences. Any changes occurs in the chromosome, sudden change. The changes may be induced or simultaneous. So, sudden spontaneous changes or this induced change occurs in the nucleotide. There are different types of mutation, point mutation and chromosomal mutation. If any mutation occurs in a particular gene or individual gene, single gene or single nucleotide, any mutation takes place, any sudden changes takes place, that is called point mutation. Point mutation. And the structure and number of chromosomes, if mutation takes place in the structure and number, is called as chromosomal mutation. Chromosomal mutation. So, mutation, which is broadly classified into two different types, point mutation and chromosomal mutation. So, this genetic variation among individuals which provides the raw material for evolution, ultimate source of evolution. So, mutation, it leads evolution. So, it is the ultimate source of evolution. So, this sudden change occurs in the chromosome, either in the chromosome or in the genes, in the structure of the chromosome or number. Number, you know that there are different, there is a definite set of chromosomes present in an organism. If any alterations occurs in the chromosome, it is called mutation, sudden change. May be spontaneous or induced. It may be induced by chemically or environmental conditions. So, if any mutation caused by agents, they are called as mutagenic agents. So, this mutation, the term mutation was introduced by Hugo de Vries in Oenothera lamarckiana. The plant name is introduced in Oenothera lamarckiana. Oenothera lamarckiana. The common name is primrose, evening primrose. Oenothera lamarckiana. So, Hugo de Vries explained in the year 1901. So, mutation definition, first you will know what is mutation. Sudden change occurs in the nucleotide sequences of the chromosome. So, within the chromosome the genes are located in different locus. So, this is broadly classified into point mutation and chromosomal mutation. You understand that point, in a particular point, in a single nucleotide sequences, any changes occurs, it is called point mutation. The entire chromosome, maybe the structure of the chromosome or the number of the chromosome, because you know the number is constant. There is a definite set of chromosome. If any changes in the occurs, if deletion or addition, so how the number changes, sometimes the chromosome may be deleted. For example, in this chromosome A, B, C, D. So, in this gene, if anything deleted, some sequences can be deleted, this one is deleted, or instead of this A, B, C, D, one more D is occur. So, addition, deletion or addition, transition or substitution. Substitute, instead of purine for pyrimidine, instead of pyrimidine for purine, substitution mutation. So, different types of mutations are there. In the next class, let us see the types of mutation in brief. So, the production of mutants. So, what happens? New varieties are formed, mutants. The name mutant. Mutants are formed by mutagenic agents. So, what are mutants? Mutants are the new variety or new form, new character. Mutants are developed by means of mutagenic agents or called mutagenesis. The process is called, by forming mutants by mutagenic agents is called mutagenesis, and the organism developed is called as mutagenized. Four terms are there. Mutants. What is mutant? During mutation, mutants are formed. The new characters are formed, mutants, through the exposure of mutagen. What is mutagen? Mutagenic agents, mutagens, and the process is called as mutagenized, the mutagenesis, and the organism, by this method the organism is developed is called mutagenized. These four terms you remember. So, once again recall what you learned today. Only 3 questions: self-sterility in nicotiana, sex determination in maize, and mutation, just introduction. Next class you are going to learn types of mutation in detail.